Okay, so in this question, you're being asked to do the reverse of what you just finished. That is, instead of taking an alignment and drawing structures, you're being given structures and asked to do the alignment. So here are 5 structures. Let's align them. Remember that you're aligning them based on the structures. You can ignore the sequence until you get to individual little bits. So let's start with this one. We have reading 5' to 3', C, U, U, C. This is the secondary structure then, just drawing the parentheses, because these are paired to something downstream. We have G, A, A, A. Those are not paired to anything. You can either put dots or dashes in there, it doesn't matter. And then, what is that? G, 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 paired upstream, U. So this is the secondary structure of this thing, drawn basically in a different format, but it's the same secondary structure. Let's have a look at the next one. Well, it's also got 4 base pairs in the helix, so we'll just lay those in. Here's the structure right there. We'll go G, A, C, G. Those are the base pairs. Here are the things it's paired to. C, G, U, C. The loop is one bigger, so we're going to have to put that in there and align it somehow. And maybe the G and the A work, so that's fine. We have UCC, and there's no way of knowing where those are going to fit in. So UCC, and I'll just arbitrarily put in a gap like so. Now we may be able to refine that a little bit later. And then at the beginning, we have an A not present in the other structure. And at the end, instead of having a single U, we have a UA. These are both U's, so I'll put it here. If this had been an A, I would probably put the gap here and put the A's together. So that one's done. Now let's add this next one. This one's a little more complicated, but it's basically the same process. Here we have a leading A, as in this one. We have 4 base pairs in a helix, G, A, C, G. In fact, it's the same sequence. But on this side, we have a bulged A in the middle of that helix. So what should that do? Well, let's put in the base pairs, C, G, U, C, but there's an A right here. And so this A goes in there because it's the sequence. That's the sequence. Here's the A. We put a gap in there because that's what a gap means, that there is no such base in these 2 sequences, and so those are gaps in the alignment, and you have the bulged A. And then you have a UA tail, same as the other, and we only have to deal now with the loop. This loop is a 4 base loop. I look at this, I see UACG, UACG. I might want to put that UACG. This, you've got purines here, variable position. You might be able to adjust that a little bit, but here you've got to conserve purine and variable positions. This is a little tough on the loops, and you might have to make some arbitrary decisions. You also might want to come back and revisit that once you've got the other ones in there. Here we have a 6 loop, so this is, or excuse me, a 5 base pair loop, and so this is a little bit longer than the other 2. I'm going to have to add some nucleotides. Where should I add them? Traditionally, you would start to consider the base to be the important part of the RNA of a helix and the loop stuff to stick out. And so I'm going to make this the extra base pair. So I'll start here at the beginning. There's no leading A, so I put a gap, CC, UC, CC, UC, and there's a G, which is a gap in the other structures. And I'll draw that as paired because, of course, it is paired to something downstream. On this side here, then, I've got to put in the corresponding sequence, and then the same as the others, G, A. There is no bulged A. G, G, and there's a trailing A. And I'll put in a gap. This leaves me with only the loop. We have C, U, U, G. Okay, so what am I going to do with that? C, U, U, G, A, C. This is where it would be nice to have these on a piece of graph paper. I would probably align those with the U, A, C, G, C, U, U, G, like so. So that the purines at the end are aligned, and then you've got pyrimidines in front of that. Next you have the last one. The last one is fairly simple. It's a simple 3-base pair stem, 3-loop, and a tail. Much simpler than the others, and so there's going to be a lot of gaps in it. We'll start at the base of the helix again. Put in the sequence C, U, U. The next two are going to be gaps because there's no such base pair for here or here. This base pair corresponds to this one to that one, and this base pair corresponds to that one. And so all these nucleotides are going to be gaps. There's a 3-loop. Because I've been putting the purine here at the back, I'm going to do that. So that I've got a block of pyrimidines in the middle, except for the first sequence, which I can't do that with anyway because it's all purines. Once again, I've got to add those gaps for these two bases here. I've also got to add the gap for that bulged A, and I end the helix with A, excuse me. The A is here. Here's the bulged A, the A, G, and then there's a UA tail. And once again, we're done.